Membrane structure and function. The plasma membrane of the cell has several important functions: structural support, regulation of what moves in and out of the cell, recognition between cells, communication between cells, and binding of cells to other cells to form tissues and organs. Diagrams of the cell membrane that we see in books often give the wrong impression of the membrane, portraying it as a solid, rigid structure. However, according to the fluid mosaic model, the membrane is a fluid structure with a mosaic of various molecules embedded in or attached to a phospholipid bilayer. Generally speaking, the fluidity of a membrane is similar to the fluidity of an oily salad dressing. Many of the molecules can move laterally within the phospholipid bilayer, much like ice cubes floating in a punch bowl, although some are attached to the cytoskeleton. The phospholipids that make up the bilayer are amphipathic, meaning they have both a hydrophilic and a hydrophobic region. The head of the phospholipid is charged and therefore hydrophilic, whereas the hydrocarbon tails are hydrophobic. When phospholipids are added to water, they self-assemble into this bilayer, which shields their hydrophobic fatty acid tails from water. The fluidity of the membrane depends on the number of saturated versus unsaturated hydrocarbons and can vary greatly in different organisms. For example, organisms that live in colder environments have membranes with a greater proportion of unsaturated fatty acids, which keeps their membranes fluid even at low temperatures. Some cold-tolerant plants can actually increase the number of unsaturated fatty acids in their membranes as the temperature drops. This change takes time, however, which is why a sudden cold spell can kill even frost-tolerant plants, something we're all too familiar with here in Colorado. Another factor that influences the fluidity of the membrane is the presence of cholesterol. Interestingly, cholesterol can serve to either increase or decrease membrane fluidity, depending on the temperature. At low temperatures, cholesterol prevents the phospholipids from packing together too tightly, increasing fluidity. At higher temperatures, cholesterol adds rigidity to the membrane, decreasing fluidity. Thus, it can be thought of as a sort of fluidity buffer for the membrane, helping it resist changes in fluidity due to fluctuations in temperature. Many of the functions of the plasma membrane are carried out by membrane proteins. Membrane proteins can be either embedded in the bilayer or attached to the cytoskeleton or fibers of the extracellular matrix. Proteins that are embedded in the bilayer are called integral proteins. Proteins attached to the inner or outer surface of the membrane are called peripheral proteins. Integral proteins oftentimes go all the way across the membrane, in which case they can be referred to as transmembrane proteins. Transmembrane proteins must be amphipathic, with hydrophobic amino acids being found in the region that crosses the lipid portion of the bilayer, and hydrophilic amino acids being located in the inner and outer leaflets of the bilayer. The term membrane-sidedness refers to the fact that the membrane has distinct inner and outer faces. The lipid layers differ in their composition from one side to the other, and each protein has a directional orientation with respect to the membrane. Major functions of membrane proteins are transport, enzymatic activity, signal transduction, cell-to-cell recognition, intercellular joining, and aiding in cell shape and structure, for example, by attaching to the cytoskeleton or to the extracellular matrix. If you have any questions about any of this content, please don't hesitate to contact me.